Thanks. Hi, everybody. We have been listening many great topics related to the archaeologies, and then right now I'm going to take you to the soil science, something cover up the things your archaeologists try to find out. Yes, I'm sorry. And then I'm from biogeography science. Then this is my study. The things I'm going to talk about is going to about what is the predictive soil mapping, what is the DMs, the data I'm using for the study, and then also about my study site, and then the methodologies, and then the result, and then a little bit discussion on the uncertainties. And then why the purpose of this study is partially a large concept about the predictive soil mapping. The things I'm talking about today is in order to use this portion of study, try to improve the accuracy in our predictive soil map. Okay, oh, sorry, I think this is the wrong slide. This is not the one.
You do the wrong way.
Okay, this is not my final slide, but we just use the one I'm using then. Yeah. Okay, this is a traditional soil map looks like, and then it's a paper made, and then they have the colorful features indicate the polygons for each soil classifier. And then for our predictive soil mapping, we're using many digital terrain models. And then the things, one thing embedded with the digital terrain models are the noise and uncertainties. And then for our study site is the Lower Fraser Valley. That's the, or another name is the Great Vancouver area. And then what I'm trying to do in the methodology is using the data extracted from the soil legacy data point are the one we have seen before, the traditional soil point. And then we're using the topography indices derived, calculated from the DMs. For my study, there are so many resolutions, but then for my study, I'm using 75 meter DMs. That's indicated cell size is 75 meters per 75 meters. And then I'm using the 4 comparisons, our original unfiltered the DMs, or using 3x3 windows filtered DMs, and then the 5x5, 7x7. After extract the training data, I run my training data to the random forest, that is statistical decision tree model. What random forest do is systematically separate training data and then the topography covariance into different groups, and then randomly select the point from each group, and then randomly select topography indices to split those points, and then to make a decision by the end for the purpose of my study is looking for the soils in the gray group. And then I'm looking at outback arrows, and then that's the comparison in my result. Okay, here is the topography indices I have been talking about. Actually, from the DM, I can derive more than 30 of them, but the reason I'm only choosing these 8, because from my previous study, these 8 are strongly correlated to describing the soil in the landscape. And then here's one of my result. We can mostly focusing on the table on the bottom, and then from the original DMs, we can tell the outback arrow is 34%, almost 35. And then for the even after filter by 7x7 windows, we have only have 31.58. It's really small amount of decreasing the outback arrow, but does give us some indicator saying this method is working. And then this is the predictive soil map by the end, but I want to show you the result indicates into each group. We can see there are some group, for some of the predictions in the gray group in the soils have a little bit increasing the accuracy, but then for some of them, for example, in the first one, the district Brunusol, there doesn't have a change at all. One reason for that is either we have a really small data point set, it doesn't really change our random forest model that much, influence that much, or the development of this soil are not really strong correlated with the environmental indices. Okay, and then here's the discussion questions. We still have some limitations. There is from our training data, because from the old soil maps, we have unbalanced sample size, we have limited soil types, And then for other limitation on the environmental covariance is we only use the topography indices. There are still other things like vegetations and so on. And then thank you for my supervisor, and then for people working in our lab, and then also for some other undergrad students who helped me through this project. And then that's the end of my last undergrad presentation. Thank you.